Welcome, and thank you for joining us today for the webinar on LiFE, an international consortium approach to researching a rare cancer syndrome. My name is Jennifer Leidenberger, and I will be your WebEx host. Before we get started, I'd like to make a few comments. All lines have been muted upon entry and will remain muted for the duration of the webinar. Please submit your questions throughout the presentation in the chat panel and select host or all panelists from the drop-down. We will ask them on your behalf during the Q&A portion of the webinar. You'll see a link in the chat box shortly if you need live closed captioning. And I'd also like to note that this webinar is being recorded. And now I'd like to turn it over to Nonike Shelburne to introduce our speakers.
Thank you, Jennifer. Again, my name is Nonike Shelburne. I'm a program director with the Epidemiology and Genomics Research Program, and also the NCA liaison to the Li-Fraumeni Exploration Consortium, which we'll refer to as LiFE throughout the presentation. Li-Fraumeni Syndrome is rare. It's an inherited cancer syndrome requiring collaborative science to address many of the needs of the growing population. The LiFE Consortium was established in 2011 to foster communication among Li-Fraumeni Syndrome investigators and to promote collaborative multi-site research to improve our understanding of Li-Fraumeni Syndrome and clinical care. Recognizing the benefit of data coordinating centers as a tool for collaborative cancer epidemiology research, our program here at NCI piloted a data coordinating center contract in 2014. The goal of the contract was to demonstrate data pooling, harmonization, management, distribution, and public and secure access through a collaborative process across cancer epidemiology consortia. And we chose one focused on rare cancer, for which LiFE was selected. During 2014 and 2015, the LiFE Consortia investigators and the Maryland-based contractor Enterprise Science Computing, or ESAC for short, created the Data Coordinating Center. Once fully operational, the Data Coordinating Center was transferred from the contractor to the consortia, and the City of Hope received that Data Coordinating Center in 2016 and has been managing it since. It is my privilege to have Dr. Jeffrey Weiss and Ms. Sharon Sand from the City of Hope here with us today to discuss the Li-Fraumeni Data Coordinating Center work to date, including challenges, successes, outcomes, and practical tips for running and housing a rare cancer data coordinating center. Dr. Jeffrey Weiss is a physician and a researcher in clinical cancer genomics, working with populations at increased risk for developing cancer because of family history or personal risk factors. He's been a member of the City of Hope team since 1996 and is expanding the reach of low-cost genetic screening materials, as well as training to doctors and nurses in underserved populations in Peru, Colombia, and Mexico. Dr. Weiss is a founding member of the Li-Fraumeni Exploration Consortium. We also have Sharon Sand with us. She is a highly experienced clinical research administrator and supervises the research core staff in the Clinical Cancer Genomics Division at City of Hope. She is administrator of the Clinical Cancer Genomics Community Research Network, managing clinical data, pedigrees, and DNA samples from the multi-country initiative. Ms. Sand was vital to the development of the Li-Fraumeni Data Coordinating Center as a member of the technical working group, has been the lead coordinator of the LiFE Consortium database since it was transferred to City of Hope in 2016. At this time, I'll turn the presentation over to Dr. Weiss and Ms. Sand.
All right. Thank you very much, and thank you for that warm introduction. So can you see me share my desktop? Share it. All right, there we go. And go ahead and go into presentation mode. And just want to confirm, Nonike, that you guys can see our screen.
We can. Thank you.
Perfect. All right, great. Well, again, this is, when we were asked to do this, Sharon and I were delighted. This is a labor of love, as really all good science is, I think. And we'll tell a story, both a little bit of a story about the syndrome, but really the big story about how do people come together to work on such an important area. So we're, as mentioned, at the City of Hope out here in Los Angeles. We're just getting to the end of our gloating season in terms of weather, but you know, it's nice 280 days a year. So we Can't argue. So let me go forward and just go to the next slide. So first off, who are Li and Fraumeni? So Frederick Li and Joseph Fraumeni Jr., many of you know about this, and for those of you who are aficionados and really know about this, that it was really extraordinary science. It was the science of observation. Really, they said, boy, there's a cluster of cancers here that is more than we expected. Rhabdomyosarcoma in children, epidemiologic study, and identification of a familial cancer syndrome. And it was 1969, so that was 50 years ago, and in the JNCI, and it was really identification of a syndrome. Now, Fred worked from Boston at the Dana-Farber Cancer Institute, but was a member of the NCI. He passed away in 2015 after a long and really amazing, illustrious career. I've had the great honor and look forward to that honor many times over of meeting with Dr. Fraumeni Jr. We've seen him, and I know they just had a seminar at the AACR, which was commemorating the syndrome. And so I'm really, really delighted to be involved in this, and I'm really honored to be a part of the program. So as a chronology, though, and for a rare disorder, and especially one that involves counting on perhaps 2 hands or 2 hands and 2 feet the number of cases, let's talk about what the syndrome looks like first. So this is actually from the ASCO core curriculum looking at teaching about Li-Fraumeni syndrome. And as you can see, it's a autosomal dominant syndrome. We know now it's associated with P53, but that wasn't always the case. And breast cancer was one of the manifestations that I would see as an adult oncologist, but really with soft tissue sarcomas and osteosarcomas in children, brain tumors in children and adults. Leukemia was thought to be an important part of the syndrome, but really was made into a secondary characteristic many years later. So the core cancers, as they're called, brain tumors, especially choroid plexus tumors in childhood, sarcomas, especially rhabdoid and liposarcomas, specifically certain types of sarcomas with known translocations and things like that are not part of the syndrome, adrenocortical carcinoma with a susceptibility window during childhood, major manifestation often being hirsutism, so you have to be adept at understanding this. Breast cancer, so you're obviously covering kind of like every tissue in the body, all the scary stuff like the brain tumors and things like that. And if a woman gets to adulthood, we're talking about breast cancer, and these are the youngest onset breast cancers I've ever seen. So not uncommon at all for a woman to have breast cancer in her 20s or early 30s. Men are carriers as well as women, so this is autosomal on chromosome 17, but I think the penetrance is less in men, and those are some of the things we still need to understand. Now, what about the ontogeny and how we learned about this? So, 1969 was the first observation, and then look at this. You're getting a paper in Cancer Research in 1988 on 24 families. That was the entire base upon they're working with. Fred Li was the lead author here, Joseph Fraumeni Jr. second author, John Mulvihill, a geneticist who's been doing this for a long time, one of the earliest people doing it. Margaret Tucker later focused more on melanoma, but this is a really strong crew at the NCI that really are the founders, in my mind, of familial cancer care. Then look at this. A few years later, a follow-up study of the same 24 families with my dear friend and colleague Judy Garber as the lead author. And this was around the time of her training, or finishing of her training, and being engaged in the syndrome. And she worked very closely with Fred Li, who was at the Farber. It wasn't until 1990 that the gene P53 was connected then to this syndrome. So this was really an interesting time. I will share with you that was when I was a third-year Med-Onc fellow, just finishing my fellowship, and it was things like P53 being linked to Li-Fraumeni syndrome. If you remember, 1990 was the year that BRCA1 was localized to chromosome 17. These were exciting times, and they still are today, but it really changed my life in the directions I went. Now, how do you recognize this beyond the pedigree that I showed you? And so there were some would call the Chompré criteria, which really sort of picked out the key features. Very early onset cancers, breast cancers under a certain age, sarcomas, and then tried to sort of do a calculated sensitivity for how do you know who to do testing on, because frankly, testing was very expensive and in fact wasn't even available commercially until Interestingly, it was the City of Hope Molecular Diagnostic Laboratory, then under the direction of Steve Sommer, who started it. He was somewhat of a maven in P53 research himself. And so City of Hope was one of the first places to offer commercial testing in the mid-2000s, so around 2005. And I'll talk about some of that data. But here we are now, 50 years from the initial definition of the syndrome. And by the way, the Champré criteria look at Li-Fraumeni, Li-Fraumeni-like. So what was the syndromic definition versus who else should be referred? We're 40 years from the discovery of the tumor protein and 30 years from the association of TP53 with Li-Fraumeni syndrome. So these are kind of interesting decades in terms of our timing for having this webinar today and for talking about a consortium focused on this disease. So this was our paper from 2009, which was a summary of the 500 or so cases who were referred for single gene testing for P53. And one of the things about this table, and this is what our contribution was at that time, was to sort of put it, if you have a core cancer under a certain age, what's the likelihood that you would have a P53 mutation? And I was sort of mirroring the BRCA criteria of the time. And so, but one of the things that came from this paper was that we saw that for women under 30 with breast cancer who were BRCA negative, that alone had a yield of about 7% for having a TP53 pathogenic variant. And that was then incorporated in the NCCN guideline. And so more women got access to testing. And really the recognition of it as a cause of early onset breast cancer, even if you didn't see the childhood cancers. First glimpse, if you will, that there might be some differences in the phenotypes across families. Second was it really did focus on the fact that we didn't find a P53 mutation in anybody who didn't have at least one of the core cancers at a young age. So those were two things. Now that's reinforced. This is just another way of looking at the data that we had from our paper. Again, 91 of 500 individuals had a mutation, and you can see the distribution of the cancers among those who were carriers. There were multiple other cancers in the series, but it was really focusing on the core cancers as sort of a dogma to be thinking about when you're thinking about the syndrome. This was a paper from the French group. The French have also one of the largest experiences. But if you look here, this is from a paper in 2015, and you'll notice that all the total N of individuals that they were following at that point were 132 kids and 219 adults, okay? So this is an entire country's consortium looking at P53, and look at the limited numbers. But what I wanted to highlight here was for the childhood presentation, the core cancers, exactly what I just cited for you. And for the adult presentation, we're talking about the breast cancers and the soft tissue sarcomas. And then lung cancer, interestingly, is one of the, among the common adult cancers, that's one of the more common ones. And you'll see the others here. Colorectal also comes in, and often at a very early onset. So this is work done at the Dana-Farber, Safna Singles group published on this, and the idea that we do screen for colon cancer starting in the 20s for these individuals. But nonetheless, the syndrome and its phenotypic features have remained remarkably durable over time. But so what was going on up to that point? So we have multiple investigators working on Li-Fraumeni, this very rare syndrome, each with their own research registry. There are relatively small numbers of patients. I showed you the two papers that were on 24 families. Most of our collaborations were one-on-one or one and a few collaborators. These are wonderful people. I am just so honored to be around with Louise Strong, David Malkin, Judy Korber, all the folks that are really well known in this realm. Great friendships, good collegial relationships, but no formal agreement on common interests in a mission. We had limited genomic tools, and testing was expensive, and there were limited commercial testing options. City of Hope was the only lab that offered single gene testing for quite a long time until the era of multi-gene panels started to kick in around 2013. With regard to advocacy, there are grateful patient advocacy at the local level. So David's got folks, Louise has got folks who are really grateful and so essential, but not really organized in a way that was going to be systemic. So that was the genesis of the consortium. First, Genesis meeting, which was in 2010 on the campus at the National Cancer Institute. I was again honored to be there, to be part of that group. And what was unique about this, and I don't want anybody to underestimate the importance and the power of this. We didn't do it with the intent that we were going to be a bunch of poobahs and academics and that this is the way of the world. But we brought with us and invited, fully embraced our partners who are our patients. And so I had some of my patients who were there. And by the way, because these families are so rare and they gravitate to centers of excellence, I share families with Judy. I share families with Kate Nathanson at UPenn. I share families with the folks at the NCI, with Dr. Savage and others. So we all had some families there, and they built up an advocacy group, a lay organization at the same time as we grew up as a consortium. And the idea was that we were joined at the hip from the start, where they can help with fundraising and awareness, and they keep our feet to the fire with the burning clinical questions that need to be answered desperately for their families. And I got to tell you, we listen to patients all the time, and I could watch and cry at many of the talks and how heartfelt they were. And we never lose touch with that. I think one of the privileges of being in the prevention realm is we're dealing with the future and how do we prevent cancer. And I've always felt privileged to be there. But I am moved today, and there's nothing more sincere and authentic than what their real experiences are. So this was the marriage. That paper that I'm citing here, Fong Mai led the paper. Fong trained with us and was at the NCI when she wrote this up for our group. You can see all the authors there really reflect the folks of the day who are some of the leaders in this realm and have been involved in P53 for a long time. And obviously it was really an honor to have the other founders there. But either way, the point was is that at the end of this paper, and most people don't see it, there was a 2-page statement or a resolution of what we were. And by the way, the acronym, Li-Fraumeni Exploration Consortium, I like to say, so we did one paper on referral guidelines, but I really haven't been a maven of P53 like my colleagues like Judy and Louise Strong and some of these real lions of that field. But I did come up with the acronym. I'll take credit for it. And I put a little light bulb on the top of the I. And so I couldn't think of a better acronym for something that is so forward-looking and prevention-oriented than LiFE. And it honored the founders. And oftentimes they have to be really strained to make an acronym. This one fell together, and I put a little light bulb, you'll see it on the emblem, on the top of the eye as a symbol of really bright light and also illuminating ideas. So, but you can read through this. I believe this will be available as a recording. So, but it was really talking about the fact that we're multidisciplinary, so many people involved, unite resources. We're talking about psychologists, researchers, genetic counselors, especially the genetic counselors and other members of this community that we want to combat and understand this disorder, and our goal is to eliminate suffering. That you can put on any medical mantra, but really here, these families do suffer. Recognize the complexity, we acknowledge that no single institution, and the rarity. And again, this goes to the rare diseases webinar concept here, that when you have a rare disorder, you have to collaborate, and put your eggs in the same basket if you want sample size and to be able to give some effective and reproducible work. So, all right, no single institution can do it. That to really benefit patients, we need to understand and work together. So we want to pool ideas, resources, and energy as the most effective means, therefore the consortium. And further, we wanted to reflect the hope and belief that cancers ultimately associated with the LFS are in many cases manageable, increasingly curable, and ultimately preventable. It goes on. I actually cut out a few paragraphs. It's a 2-page manifesto. But LiFE will work to understand the mechanisms of disorder, predict individual cancer risk, detect cancers at their earliest stage and most treatable stage, develop new therapeutic strategies, and support affected patients and their families. And that will operate in partnership with the LFSA, the Li-Fraumeni Syndrome Association, and other advocacy groups and international organizations and funding agencies to deliver on our promise. So this is really, again, a unique concept of putting together the consortium of academics with the group of the lay individuals who will be the fire under our feet or the light on our lamp to help us not lose the way on the way. And so again, I'm really honored to be part of that. And it's at this point I'm going to transfer the control to Sharon, and she'll talk a little bit about sort of the nuts and bolts of who's involved, what were the first iterations of the database, and then some transitions that were unique to our experience at the City of Hope. Sharon.
Well, hello there. I'm honored to be with you here today and to share our experience with you about establishing the Data Coordinating Center here at City of Hope, and also being part of the initial startup of the Data Coordinating Center. So the participating sites included these initially, and is actually continuing to grow pretty rapidly right now. But we have Maria Isabel Achaz from Brazil, who's been involved from the beginning, Gareth Evans from the UK, ourselves, Judy Garber from the Dana-Farber, Kim Nichols at St. Jude, Dr. Kratz from Germany, and then Dr. Savage from the NIH, Louis Strong from MD Anderson, and Josh Schiffman from University of Utah, and David Malkin, who is in Canada, a hospital for sick children. So this was the initial steering committee as it was set up, and it has been evolving a little bit. We'll talk about that a little bit later. So as Nanike mentioned, the NCI contracted with Enterprise Science and Computing, or ESAC as we'll refer to them, to develop the LiFE Data Coordinating Center as a model. And they developed and documented the DCC development for LiFE. They did this with input from the steering committee and also the technical working group, which was convened primarily with clinical research administrators such as myself from each of the principal investigator sites. And at the end of that 2-year period, a technical paper was drafted. So then once that process had been completed, it was time to turn over the Data Coordinating Center to one of the steering committee sites, and that ended up being the City of Hope. So we took it over, and the Li-Fraumeni Syndrome Association, which is the advocacy group started in parallel with LiFE, provided partial funding for a coordinator, which really helped to get us going. So in the first iteration of the Data Coordinating Center database setup, ESAC established this fairly elaborate system that would take data from each of the participating sites in like an Excel format and load that data, validate it, and harmonize it. At that point, it would go into the database, and reports would be automatically generated through a system that was a dynamic system and could be seen via a website by the PIs and administrators, but also in limited fashion by public users. And the reason for this was so that people could see what was going on with LiFE, and also so that other researchers who were not part of the steering committee or part of LiFE could see the type of data that was available for research and could submit proposals and query the steering committee as to whether they could use the data for research. So this was one of the pages that was part of that dynamic database demonstration, and was a great representation of the data. A little bit challenged with data harmonization, which is what the technical working group was working on, and I'll show you an example of one of the challenges later. We had so many different sites collecting basically the same information, but all in slightly different ways. And then within the website and in the database, we were able to see the data in several different ways, both by genetic status, by the type of cancer that was represented in families and by sites, and whether biospecimens were available by different types of cancer and genetic status. So now we have the database has been growing fairly quickly, and we have assembled nearly about 1169 TP53 carriers in over 513 families. So this is a representation of the cases that are TP53 positive, and then by age at first diagnosis. And you can see here the early childhood cancers followed by later childhood, early adult, and then later adulthood cancers. Then we have the data separated by positive for TP53, negative for TP53, and then variants of uncertain significance. The negatives are important for us to be able to understand the penetrance and variability within families, as well as the probability calculations that can be done with calculating for TP53. Variants of uncertain significance are those variants that show up in TP53, but we don't know whether they're cancer causing. And important work is being done in this area to adjudicate whether those are truly positive or truly negative. So then this is just a quick representation of data assembled by site, and this is continuing to grow. As we've recently transitioned our database, we've been able to increase these numbers, both in terms of numbers by site and also the numbers of sites that are contributing. And then this is a breakdown of the cases by, or the families by country. So primarily the US, but then followed by Brazil, the UK, and then several others. This is a representation of the cases broken down by positive cases, both affected and unaffected. And affected means they have cancer, and then unaffected means they don't have cancer. And we're able to do probability studies with this data because we have probability and penetrance studies because we have age for each of the individuals. So we can see if they're, age 30 and haven't gotten cancer, or age 50 and haven't gotten cancer and have a TP53 variant. So for race and ethnicity, the vast majority is white, followed by a fairly significant representation of Hispanic. Then the other categories are not as well represented, but we're anticipating that with the growth of both in sites and then as well as an increase in numbers from especially the sites that have more diversity, we'll be able to increase this diversity. So for the logistics, we have a monthly steering committee meeting, and that's done by phone, and it includes the LFSA leaders as well. So the advocacy group is represented on those calls. We also have semi-annual, so every 2 years we have a LiFE and LFSA meeting, and those are great meetings which include the researchers, where we have steering committee meetings, but also the families, so educational events and sessions for the families in conjunction with the researchers. So in terms of logistics, we have review of research proposals and implementation of data use agreements. We're just getting started with this and pretty excited that we're able to provide research data for collaborating sites who have proposed ideas. And because of this impetus, the database has continued to grow both in depth and breadth. The data transfer agreements are set up with each site before any data is collected, and then the data collection begins. So some observations and limitations. So although with the setup at ESAC, the summary statistics and graphics were very aesthetically pleasing. It was a beautiful website and a way to present the data so that people can see it. The visit statistics were not exciting, and we didn't, unfortunately, generate much in the way of research proposals. So we also were not getting new data from the member sites because so much of the effort was put into establishing the logistics of that process. So we also had the challenge of adding additional fields. So once the data dictionary was set, it was difficult to add those additional fields when the focus was on the data harmonization. The data dictionary was set up at ESAC with the steering committee and the technical working group at the beginning, and we've been able to expand that recently. So the variant level data was not included initially, but we've been able to add that and collect data from most of the sites on the variant level data for each of their cases recently. We also added breast cancer hormone receptor status, so ER/PR HER2 data. That was recently added as well, and that was in direct response to a research proposal that was looking at TP53 cases and hormone receptor status based on TP53 status. In the original database, it was quite complete, but there was only categorical or limited family history data and no pedigrees. So no family history representation. The steering committee decided to transfer the data to a pedigree level relational database, and the reason they did that was because they wanted to be able to view the pedigree level data. We wanted to be able to include the specific TP53 variant data, and we also wanted to be able to give sites access to their data for submission and also for updates. So by doing this, we were able to create better user operability on our side and ability to add and revise data fields as we realized if they were too constraining or not broad enough. And then we also expanded the phenotypic data. And the reason we were able to do this was because we've been coordinating the Clinical Cancer Genomics Community Research Network for over 20 years and using the Progeny Relational Pedigree database for that consortium pretty much the entire time. And that consortium is 45 sites across the US and in Latin America with over 25,000 pedigrees and DNA data. We also use the same backbone to coordinate another consortium, Enigma, which is another international consortium. So some of the challenges that we've had since taking on the Data Coordinating Center, one is funding. While we have had very generous funding from the Li-Fraumeni Syndrome Association, it does not cover all the costs that we have for managing the database and coordinating the data transfer and updates and all of that, including the informatics person, the administrator, and the clinical research assistant. Another challenge has been defining roles, the membership of LiFE, and the steering committee, because it was very clear who that was when it started, but is starting to evolve a little bit. And we want to make room for new folks and make sure that it's clear which people are in which roles. The data dictionary has been a little bit of a challenge. We've been working on refining that, clarifying that, and then also expanding that. Data harmonization, which I'll show you an example of in a minute, has been challenging because there's so many different ways of representing a specific cancer, or even a genetic variant might be in two different types of nomenclature. And then with an international consortium, there's been some barriers with some data transfer agreements, legal types of issues from different organizations and mostly from international sites. So Also, the timing of the steering committee song conferences, we have to make it when everybody's awake, and somehow we managed to do that. So for data harmonization, this is just a quick example of a very small part of the data. So sarcoma is listed here as what we've mapped, and these are the types of data that were submitted. So all sarcoma, but slightly different in terms of how they're worded. And then osteosarcoma, all of these down here, these are, we had to realize that these were all osteosarcoma and then map them so that we can categorize them into a reasonable number of categories. So lessons learned, it's important to define everything as much as possible up front, but to continue to be flexible to do that going forward as well. And this is especially important for the data dictionary, but also to plan for the research proposals, including what the authorship rules will be if research proposals are granted. And then we talked about flexibility to add more fields. That's important as you, like we did, we got the research proposal that had data we didn't have collected. But by being fairly nimble, we were able to collect that additional data. And then plan for funding. It's important to have sustainability and continuity plans to be able to continue the coordination. And then data submission, timing, and method. So it's pretty straightforward to collect the first cases, but then what do you do when somebody has an update to their case? How do you know what's new data, and how do you identify and or collate that data into the existing data? And then how do you add new data, so new cases or new individuals on a pedigree? And I'll hand it back to Dr. Weiss here.
Great. So actually the key ingredient on that last list is Sharon Sand. So no, people like Sharon. So smart, critical, analytical. You got people like me and Judy Garber biting off way more than we can chew. We give ourselves the glad-handed title of visionary, but really we're just scattered. So Sharon and people like Sharon help to give us alignment. Alec Cervantes is the one who's helping to organize our steering committee calls. And so we really rely on folks to help us with that structure, but you need leadership and you need structure. And both of those are going to be important for your success down the line. So now I want to talk about, so if we had this consortium 10 years ago, and it was a well-organized data coordinating center and well-supported, again, when it comes to that data coordinating center role that we took on, it was really that infamous movie where they have everybody in a line in the Army and they say, whoever wants to do this, step forward, and everybody else steps back. So we were like the last one standing, but we were willing to take it on because we felt really it was important, and we knew that there would be some bridging to get to where we need to be. So one of the first things that we wanted to talk about was that, okay, how does this consortium help advance our science? And so I want to give you just a few quick examples. It's still very early in the running because we really are just really hitting the ground in terms of our establishment. But first off was a wonderful, amazing study by Villani et al., and this is David Malkin's group in Toronto, and we were all waiting for a prospective study of a highly regimented surveillance regimen that had an outcome related to survival. And that was because there was just watchful waiting for most kids. We didn't even want to know whether they were carriers. Just that they started bleeding or had a lump, you did something. So that was not a way to live. But now we know that whole body MRI, rigorous screening for biochemical signs of adrenocortical carcinoma, et cetera, actually has a role. So where does the LiFE Consortium come in? That was their work. They had been doing this for some time, and it was their study that then stimulated a lot of others to do it. So the NCI, the lead program over at MD Anderson, that's Dr. Strong's group, we were following the same protocol, albeit unfunded, but followed the same protocol. And so what happened in some of our steering committee calls is we were able to put together all of the, if you will, the thought leaders in this realm, and this paper was born from that, which is a meta-analysis. There was a series of about 4 papers that came out demonstrating the yield of whole body MRI, and in part catalyzing the integration of whole body MRI as a insurable care for individuals with Li-Fraumeni syndrome with regard to the NCCN guideline. So you can see on this list of authors for this meta-analysis, we had maybe 20 cases in. This put together all the other cases that had their own separate funded studies. They got published separately, and then this was our meta-analysis, and this was really a product. If you look in there, all the people who are on the steering committee are there. Sharon Savage, Tom's one of my colleagues, David Malkin's there, Josh Schiffman, Gareth Evans. All of us are here. So this was, I think, really, I consider it a product of the consortium, even though it didn't use the database per se. We all still had our own databases. But I want to bring that out. This is really important translational stuff. Then the second thing is that one of the first projects we did as part of a consortium, we published in 2012, Maschiari et al., this top line, under the banner of the LiFE Consortium, but really before the database had even had the data deposited in it and validated. And this was, hey, let's look at breast cancer, because the two previous studies, 2010 and 2012 from this group, had 9 and 30 cases respectively. So we're talking really small numbers, but we want to say, what's the snapshot of breast cancer in this context? And strikingly, it was HER2/neu positive. So it's triple positive as compared to triple negative. So that's what our paper said. And this table is actually from our paper that we published in 2012. This was a LiFE Consortium effort. But look at the number, 32 cases, okay? And at that point, this is the world's literature, was 71 cases of breast cancer that have been immunophenotyped to be able to know that this seems to be a dominant phenotype compared to the reported population. Well, one of our first formal concepts that's been submitted to the committee, approved by the committee, required us to go out and get new data, the receptor data, but we were able to, oops, let me do this again. So data recently organized for the new project proposal, which is going to be led by Mandy Spurtle and Christina Fortuno in Australia, was that we were able to get data strikingly on 196 cases of the 502 that are in the database. And this is on relatively short order with no money, okay? So we're not giving anybody any money to go back to their database or go back to the medical record and collect this data. But this is out of the commitment to this mission that we have. But look at that, 196 compared to the 71 from before. So this means we can do this science and we can do it better. We will get some solid data out of this. This will be a joint publication that will recognize the members. We have to sort out those rules yet, but already with an N of 196, it's a very reasonable study. They intend to use that phenotype, if they confirm the phenotype of the carriers, to help adjudicate. They're working on a model for working on VUSs in P53, so that they can use a predictive model that allows to take the phenotype, like if it's HER2 ER positive, is that predictive? They can incorporate that in the model. So there's a translational element to this, not just to recognition that it's HER2 positive, but that if that actually predicts the likelihood that mutation or that variant was pathogenic, that moves us forward too. Another project that is just getting off the ground, which we're really excited about, it's been going for a while, and this was published by Humarana at the Dana-Farber. Judy Garber's the senior author, and this was a partnership not with LiFE Consortium, but rather with Ambry Genetics. But what they published in JNCI this past year was that, boy, when we pick up P53 mutations on multi-gene panels, especially when they're qualified for something else I'm going to talk to you about, they seem to have a little bit different phenotype. And what they're showing here, and this is from the paper, was that the age at diagnosis was pushed. This is single gene testing compared to multi-gene testing, single gene versus multi-gene. So you can see that there's a slightly later age at onset if you weren't looking for Li-Fraumeni syndrome. So that must mean that the phenotype is expanded a bit. So that's one important observation. Another observation was from our own group, where I was starting to get P53s on multi-gene panel tests that I just flat out didn't believe. 58-year-old with ER-positive, HER2-negative breast cancer, just wasn't convinced the lab to work with us, and we found out that when you look at certain parameters, in this case, low minor allele fraction, so in other words, the P53 was there, it was a bona fide analytic finding, but it didn't look like a regular kind of a case from a phenotype perspective, and the allele fraction was less than the 50-50 you expect for heterozygosity. Turns out that most of these are what we call aberrant clonal expansion, and essentially analytically correct. It's in the blood, but it wasn't in the constitution of the individual. And most of those, when we did confirmatory testing, meaning we looked at other tissues, we showed that 96% of the time it was not in the other tissues. Therefore, at the least it's mosaic, and at the most it's most likely clonal hematopoiesis. So putting those two studies together, all of the P53s coming out the door we have to be careful about when they're multi-gene panel and the phenotype doesn't fit. So how do we do studies to understand the phenotype and penetrance? So we now have to qualify these folks. So we have an R01 that we wrote using our collaboration with the group, and the title is Precision Approaches to Refining TP53-Associated Cancer Risk. This will assemble the largest cohort of P53 carriers. We've embraced the consortium, so the LiFE Consortium, but in addition reached out to Geisinger, reached out to Orion, reached out to other sources of these testing processes and all of the commercial labs. So all of the major commercial labs have agreed to collaborate by at least referring these families to us via their provider so that we can then do secondary accruals within these families our goal is to accrue at least 2 to 3 persons per family in addition to the proband to do segregation analyses. And we have Chris Amos as one of our co-PIs. So it's Dr. Garber, myself, and Chris Amos, all of us representing the Li-Fraumeni Consortium. And one of the key things is if this is funded, it's got a nice score for the R01. So, from your mouth to God's ears, we'll hope that it'll be funded this summer when the council meets. But the idea is to determine the prevalence and clinical implications of aberrant clonal expansion. So what does it mean if you have clonal hematopoiesis? It could be good news you don't have Li-Fraumeni syndrome, bad news you might be at risk for leukemia. So we're going to figure that out from a prospective perspective. But once we have a cohort that's adequately sized, we expect to be able to help define penetrance with narrow confidence intervals as well as modifiers. So we'll be looking at SNPs and polygenic risk scores as modifiers of P53-associated risk. Why is there such a variation of these phenotypes? And at the end of this data is all going to be incorporated in the database and will then be available to the larger community for studies. And remember, all these consortia, the goal is future studies, not just the present. So we'll do our shameless pitch already, which is for anybody in the consortium, P53s, we need to collaborate. Anybody who's got P53 would love to have them in the consortium. Any one of the consortium members can enroll these patients, and they can then be part of this. If you have a large number of these patients, we're not necessarily a revolving door, but we're an open door to having people coming in who have some skin in the game and want to be part of our group. So this is all collegial. It's not fixed in stone. Here's the contact information for our group. So the LiFE DCC, that's at cityofhope.org. That'll get to Alec or Sharon or one of us. The contact for the Li-Fraumeni Syndrome organization. So you want help with your families. You want help to help your families find resources and to share what they are experiencing. Because as we have our annual meetings, we haven't had them, we didn't talk about that yet, but we had the meeting in DC, had a meeting in Farber at Boston, we had a meeting in Ohio. The last meeting was in Toronto last spring. The next meeting is going to be in Boston in, I believe, the fall or summer of 2020. So we're looking very much forward to that next meeting. But during those meetings, we have an LFSA meeting and the main consortium meeting. So these are two ways in which we do it. The other sense is if you propose a concept, expect that we want you to include some measure of support for the Data Coordinating Center. We gave up on sort of just support for a consortium, but rather support for projects that each incrementally help to support the Data Coordinating Center, because we really have 1/2 of 1 FTE available to us for this. The rest is all donated at this point. So, but I think it's going to be worthwhile. I think it's going to be really exciting. And that's the end of our presentation. I want to thank you all very much for your attention. If you sat in through the whole thing, we hope that it was stimulating and that you'll use this for inspiring your work with other rare disorders. Do we need to change the bouncing ball? Oh, Jennifer, you already
I already took it back from you guys. Thank you so much for the presentation. It was great. I know we do have a few questions coming in. I do want to remind our audience, please submit your questions using the chat or Q&A box and select all panelists or hosts, and our moderator will ask them on your behalf. So, Nonike, I'll pass it over to you.
So, Jeffrey and Sharon, one question that came in was, you mentioned that many of these, several patients that have rare cancers do go to various cancer centers for assessment and treatment, and you mentioned that that was a problem or something you noted within the Li-Fraumeni group. When you were creating the Data Coordinating Center, how did you identify those patients to make sure you didn't count them twice?
Yeah, so that's a great question. And to be honest, there's not a formal process yet. What we know is that what it is Judy sends them to me. I would send one back to Judy. So it was more colleague to colleague, because if the patient brings their medical records, we know who else is in the field, and so we'd recognize that, or that their extended family had members in different cities, and we would refer actively to those folks that we know, because they need specialized care. That's an informal approach. A formal approach, which we believe is now enabled, is by using a pedigree-based program. We can both look at, okay, we have 2 pedigrees that have been submitted. We can look for overlap, and there'll be bioinformatic ways for us to do that. The second way to do that is we did not originally have variant-level data, because from the perspective of at least one of our contributing centers, their RB thought that a variant, because of such a rare syndrome, would identify the family. Now that we have more than 1,000 people, that's not so much the worry. The pedigrees still are to some extent, but they're private. They're in our setting. So we can match by variant. And then among those with the same variant, we can look at the pedigree and look for structural similarity. Right. Our projects will include, or do include, genetic counselors. So Kathy Schneider and the group and others at the Dana-Farber, we've got our group is going to have Beta Net Array. So we will actively curate by looking at pedigree structure and looking at the specific variant. I think that's the best we can do.
Thank you very much.
That's short of ancestry informative markers and identity. When we do, one of the things we'll do with the project when it's funded is we'll be looking at all the SNPs, and they'll have a whole genome-wide association backbone. Clearly, you can establish identity from that. Okay.
Thank you. Another question, you mentioned a technical paper being drafted. Is that paper accessible, and really what does it include?
Yeah. That's a great question. I mean, that was originally drafted, as you know, by the folks at ESAC with our team. It went through at least one round of review at one place. Now that we've changed the database, we've changed the focus of the paper a little bit, formalizing it is being submitted. So as soon as that's available, we'll put on the website perhaps links to the appropriate literature. So hopefully we'll have a growing bibliography to share with folks. That's the other thing, is giving back to community, making sure that in our new website design, by the way, I don't know if you mentioned it, Sharon, but with Jen Perry, who's one of the leaders at the LFSA, president of the LFSA, we're hoping to maybe cohabitate the 2 websites. So have the web presence, the external facing web presence, really a stone's throw, a sister to the LFSA site, so that because we really want to continue to cultivate that interrelationship between the advocacy organization and the academic folks.
Thank you. Another question, there's a lot of standardization work going on in HL7 within the genomics work group to be able to use data directly from the electronic health record to limit harmonization efforts and increase productivity. Have you looked into this to improve data?
So it's a great question. So HL7 is basically, for those of you who are, and again, I'm only know enough to be dangerous here, I'm just a physician, which is a communication tool. Basically, it's health language, HL7 tool that basically allows us to move things. For example, you can take the Surgeon General's family history website, and patients can enter family history into that website, create their own pedigree, and there's a backdoor way to take an HL7 translator and move that actually into a pedigree drawing program. So just to give you one example, this is something when I talked to Kevin Hughes about this when he was developing his Hughes RiskGap software. So absolutely, we're thinking about that in our own world because as I started our own consortium, which aren't just P53, it's all cancer genetics, we've been doing data mining in our EMR for some time for patients who are enrolled in our registry, gave us permission to help get follow-up data. And so we need to do more of that. There are HL7 connections that are already made between family history forms in our progeny database and things like that. So yeah, the whole point is to try and make it interoperable. At our own center, we're hoping to integrate, for example, progeny or a data pedigree drawing program in the EMR for the whole institution, because nobody takes adequate pedigrees outside of the genetic counselors who we all work with, or those of us if we had the tincture of time. So I think HL7 is one of those programming languages that are translating languages. It'll be very helpful, but we are working on doing data mining to get that because that's the way of the world, right? It's the big data, the data mining. But this is a rare disorder. I will make one caveat, one comment. Brad Coffey, who's at Myriad, had done some work, presented at the American Society of Human Genetics meeting, pointed out that even 50% allele fraction P53s could still be false. And so he did this by looking at NOMAD. So NOMAD is one of those publicly available databases. And there's 2 papers out there that suggest that we just didn't appreciate how common P53 was. No, we just didn't appreciate that there were false positives in the database and that it is still rare, albeit worldwide you get enough to work with. So we think that there's conflation in the current population databases regarding P53 because this is a severe syndrome. How could they be unaffected at 80? They did the other experiment with a gene called ASXL1. It's a heme gene that's involved in leukemia, or if it's inherited as a germline trait, it's a profound developmental disorder. They don't live long. Well, it was frequent in the NOMAD database, and we're certain that reflects, again, confounding of epidemiology by these sorts of things. So I'll stop there because I could go forever on that.
All right, next question. We've established an international consortium in retinoblastoma and second cancer research last year. It proves difficult to have all legal counsels agree on the contracts, such as data sharing, data transfer, data use agreements. What are your tips for speeding this process along?
Wow, yeah, that's tough. So ESAC started the process, and in conjunction with the NCI, and we had to redo all of the data transfer agreements so that they were between City of Hope and the sites. We had our legal team in our Office of Tech Transfer was able to successfully do that. We didn't have too many hiccups.
Yeah, so that's a constant problem. Just will tell you, it's a constant problem. Internationally, it was a challenge. So, for example, for the Brazilians, there was a rule that we weren't aware of that the PI, essentially the protocol goes with the PI. So if a PI leaves an institution, it's not the institution that represents for that protocol. So that was an interesting quirk. We worked it out because they're good people, and they're going to work it out. And we have now 2 people in Brazil, one representing the old database, who's a new person, and one representing a new accrual. So there's things you have to work out. And as busy clinician scientists, no small task to get this stuff through legal. Half the time we don't even know who those folks are. But and you want to develop a close relationship with your tech transfer guy. They can be your advocate. Don't look at them as the enemy. They're just doing their job. It is a barrier-oriented process, though, and I'll tell you that even one of our major anchor tenants in the current consortium still hasn't completed their DUA. Every time it's, oh, we're going to get a new coordinator. Oh, they just changed the lawyer. So I'm sympathetic more than giving you insight.
And I will add that the National Cancer Institute does have transfer and use agreements that can be used as templates, and that was the starting point for this project. And the various centers did have some minor changes, but it was a good way to start. So should anyone on the call be interested, you can give us an email, and we can connect you with that. We're at time, but I just wanted to end with this one last question to you guys, was how can institutions not yet part of the LiFE Consortium join?
Okay, so the last slide we had sort of the contact information for this. We are not the steering committee. We are the custodians and the custodians of the galaxy here in terms of the database. But please reach out to that email address, which was, I believe, was lifedcc@coh.org. We will bring that. We organize the monthly steering committee meetings. We just had somebody, for example, from UPenn join, but we do expect that there will be some substantial clinical contribution they can make, or that will suggest that they fold in with one of the others and collaborate with somebody who's already in. They'll still be recognized as being part of the group. But please reach out to us with queries and some sort of a snapshot of what is it you have in hand or expect to have in terms of wanting to participate. We're delighted. We will make it as transparent as possible. We'll hope to have the new web presence up as well in the not too distant future to be able to make it clear. But that's the best thing to say is just reach out. We're really, like I said, we're open door in that regard. We need everybody to do this for rare disorders.
Great. Thank you so much to both of you for your great presentation, and to our audience for some great questions that came through. As a reminder, this webinar is recorded and it will be posted within the next week or so. So please continue to check back on the Rare Cancers website, as well as any upcoming events that we'll be having later this fall. So thank you, everybody.